Tejasvi, go ahead. Yes, let me just share my screen really quick. Okay, let me know if you can see that. Yes, yes. But why is it that we don't see the present button and only it says send to teacher present? Yeah, perfect. Yeah, click that. Yeah, perfect. Go ahead.
All right. So hello everybody, my name is Tejasvi Kondury and I'm a sophomore at Wakeland High School here in Frisco, Texas. And today I'll just be talking about YAP and its role in Alzheimer's. Okay, so the research topic that I've chosen to go more into is how can YAP, which is the yes-associated protein, help target astrocytic senescence in order to fight the cognitive decline present in Alzheimer's disease. And so just for background information, Alzheimer's disease is a neurodegenerative condition characterized by a loss in cognitive function and memory loss, as we all know. And it's mostly prevalent in our older population. And so there's many different causes really that help Alzheimer's progress. But one of the things that I really want to hone in on was the clumps that the beta amyloid proteins form and then hyperphosphorylated tau. And so these two proteins will clump together and form these like clumps in our brain. And so why is this important? It's because Alzheimer's disease progressively impairs cognitive function, memory, and our ability to perform daily tasks independently. And also another thing that's of great importance is worldwide, our older population continues to grow. And because Alzheimer's disease is a disease that mainly is present in an older population, it's something of significant concern to both researchers and medical professionals alike right now. So slowing down or preventing the cognitive decline could significantly improve the quality of life for individuals affected by this disease, and it would allow them to maintain their independence and engage in meaningful activities for longer periods. So with my literary review, I wanted to look at components that play a role into Alzheimer's disease with respect to YAP. And because I wanted to target astrocytic senescence and its relation to YAP, I thought it would be beneficial to get to know what astrocytes were. So astrocytes are a type of glial cell in our nervous system, and they play a crucial role in learning, memory, and synaptic plasticity. Synaptic plasticity is nothing but the ability of our synapses, which is the gap in between our neurons, and their functionality increases or decreases based on how much we use them. And so it's important to keep in mind that synaptic plasticity will probably reduce with aging as well. But what astrocytes do is that in response to the beta amyloid and tau aggregates, they initiate a senescence or deterioration pathway. And so this just contributes to further inflammation in our neurons and contributes to the further development of AD and therefore the decline in cognitive function. And so astrocytes that deteriorate will also exhibit dysregulation of their normal functions, aka the learning and memory aspects, and they'll also lead to decreased support for neuronal health. And so with respect to cognitive function in astrocytes, I came across neurogenesis. And so this is a process that happens in our hippocampus, and it's significant for brain development and cognitive function. Neurogenesis is significantly impacted by astrocytic senescence. And because neurogenesis is a process, again, that's important for cognitive function. And when astrocytes aren't able to undergo their proper functions, they stop working as they should. So this contributes again to decline in the neurogenesis process and heightens neuronal vulnerability. And so going over to YAP now, YAP is the yes-associated protein, and it's a transcriptional cofactor, which is just a byproduct of a cellular pathway. It's a cofactor of the hippo pathway. And so YAP has been shown to have an important role in tissue generation and organ regeneration, but also for the maintenance of healthy nerve cells. And so with respect to diseases like AD, YAP experiences downregulation in senescent astrocytes. However, when I was looking into it, YAP has also been shown to hinder the senescence process, and that's really what I wanted to touch base on with my research today. My methodology was just going into different research articles, surveying it, and then putting them together in a comprehensive literary journal. And so for the results, I wanted to examine a study done on mice. So hippocampal tissue samples were taken from 2-month-old compared to 24-month-old mice, just to compare the age ranges. But as expected with this, we need to look at these results here. So 24-month-old mice exhibited a downregulation in YAP as well as lamin B1, and lamin B1 is nothing but a senescence marker. So we would, if senescence is undergoing, again, we would have lower levels of lamin B1. And so to confirm if YAP is also downregulated in aging-related diseases.
Tejasvi, sorry to interrupt you. Like, are you like moving the slide forward? First, like in my, like on my computer, I can just see the first slide
Ooh,
only.
oh, pardon, my bad. It's not allowing me to resume sharing. It's saying someone paused it on my behalf. So,
Is it?
yeah,
Could you exit the presentation and
yes, my bad. I'm so sorry. Okay, are you able to see this?
Yeah, see now.
Okay. Thank you so much for letting me know. Okay, so we have a lower level of YAP in both the in just the older mice. And so when we compare it to the AD model mice, again, they have a downregulation of YAP. And so what this indicates to us is that senescence is undergoing in these mice, and they're experiencing a downregulation of YAP. And so in order to test whether YAP helps out to stabilize cognitive function, it was injected into mice via a treatment in order to stimulate YAP. And so it was tested via the Y-maze test and the Barnes-maze test. The Y-maze test is nothing but a spatial awareness test for mice. And so it tests how quickly they're able to think and assess their surroundings. And so as you can see, alternations here on the side, it's nothing but how many times are the mice able to navigate the maze without repeating it? So theoretically, you want a higher number of alternations. And so as we can see with the old mice here and also the AD model mice who were given the treatment, they have a higher level of alternations when compared to the control group. And then down here, the results of the Barnes-Maze test. And the Barnes-Maze test is another spatial learning test, but specifically designed just for rodents. And so latency with this, again, is how quickly were the mice able to navigate the test. And so this is with just respect to the AD mice. So the AD mice that were given the treatment here were able to navigate the maze a lot quicker than the mice who were not given the treatment. And so what this just goes to show us, again, is that YAP has a role in potentially helping the mice stabilize cognitive function, and they're able to retain what they're looking at a lot easier. And so this just shows us that taken together, the results of these studies prove to us that YAP could potentially help target astrocytic senescence in order to fight the cognitive decline present in AD. And this was demonstrated to us via the functionality of the aged and the AD model mice. However, YAP has also been shown to support tumor growth in various organisms, especially rodents as well. And so as with any potential treatment, it would need to be researched further in order to stabilize its role as a therapeutic intervention for Alzheimer's. And just a final thank you. I enjoyed Dr. Varghese' expertise and having a one-on-one session with him. And his lectures really helped me put together my paper and take any information that I needed. And with Professor Vogel, I loved how positive his classes were. It was just a really inclusive space for everyone to comfortably share their ideas and their progress. And I want to say thank you to the Gifted Gabber Research Program as well. I think it really helped me step out of my comfort zone and put together, such a comprehensive piece of writing that I don't think I would have pushed myself to do outside of this. And so now I know where my interests lie and where I can go from here. So thank you to everybody for listening.
Great, great job, Tejasvi. You can stop sharing. A question for Tejasvi, Dr. Varki.
excellent presentation, Tejasvi. You went like really deep into the whole mechanism and all. Like I was really impressed with what you presented here today. And I mean, so one of the things that like the questions that I would have to like ask you is like, did you also look into if there are any already some like research or something else like going in clinical trials related to YAP protein, or are they still considering um, about this thing? Did you look into clinical trials?
Yeah, so there's already various sorts of treatments that you have for AD, but with respect to YAP, there's trials with it going on for, I know, like organ regeneration, I believe. So they were looking at
Mm-hmm.
how YAP can help regenerate like liver cells and stuff like that. But it also, they've been researching it because YAP has a role in suppressing apoptotic genes, so it leads to the progression
Okay.
of cancer. So they're trying to look at if it was to be used for a therapeutic intervention, how can we prevent it from also leading to the progression of a cancer? Because we don't want that when we're trying to treat someone.
Okay, so probably like mixing it or like combining it with other drugs, which
Correct.
kind of control, like, or maybe like control the uncontrolled growth of cells.
Yes.
So, okay, good, very good.
Thank you so much.
Okay, I just want to take a moment to thank Professor Varkey, Dr. Varkey. I mean, without him, there would be no neuroscience program, right? The amount of effort, it's very hard to find a person who has the patience to mentor high school students. So a big round of applause to Dr. Varkey. Thank you so much for being a wonderful mentor. Okay, so